Hi, Sarah, do you want to say hello first?
Yes, hello. Thank you so much for inviting me to give the talk today.
Okay, thank you. Okay, let me introduce Dr. Sarah. Dr. Sarah obtained her PhD degree in 2016 from Texas A&M University with Adam Jones. After a 2-year postdoctor at National Institute at Mathematical and Biology Systems in University of Tennessee, Sarah began her current position as a lecturer in School of Biology Science in University of Canterbury in Christchurch, New Zealand. Her research group works on understanding the ways in which variation is maintained, with a particular focus on sexual dimorphic traits. She used genomics, transcriptome data to learn about the evolution of wearable traits. In addition to stimulation models and the studies of animal behavior, a fun fact about her is that since moving to New Zealand, she has learned how to whitewater kayak. Today, she will be talking about her working revealing the role of selection in the evolution of Gulf population. Okay. Let me play the video of her recording.
Tēnā koutou katoa, e te mana whenua nai tuahuriri, tēnā koutou. Welcome to my talk, everybody. Thank you for coming to this last day of Sing Bio. I'm very excited for it. And I'm going to kick off this session by talking about how I have used genomic data to understand the role of selection in the evolution of sex role reversed pipefish. Before I get into any of the actual science, I want to acknowledge how collaborative this type of research is. And I want to thank everyone who has been involved in this. So including all of my funders, plus my research group, my PhD advisor, Adam Jones, and my former lab mate, Emily Rose, both of whom contributed substantially to the research I'm going to be talking about today, in addition to all of my other collaborators. And of course, the organizers of the 4th Sing Bio conference who invited me to give this keynote speech. So, selection can drive the evolution of a wide variety of traits, and we know that it can cause rapid speciation. We only have to look to our favorite family of fishes, syngnathids, to see how this is played out with unusual body shapes and coloration patterns that allow animals to blend into their environment, to colorful, flashy display traits in many different pipefish species, plus further beautiful adornments to the bodies of these fish that also provide camouflage, as well as are likely involved in mate choice. So we know that selection can be very important in the speciation process. But we also know that selection can lead to the removal of variation from populations. In this example from Heliconius butterflies, there's selection acting on particular coloration patterns pointed to here with the arrows, and researchers have identified particular genes that are involved in the in creating those color patterns. And if we look at genetic diversity, which is what's plotted here on the bottom part on the y-axis, we can see that in those genes that are known to be involved in these strongly selected color patterns, there's a strong reduction in genetic diversity, right? So we know that selection on traits can lead to the removal of variation, both at the genetic level and at the trait level. Of course, selection is also countered by migration and mutation and can be overwhelmed by genetic drift. All of these different mechanisms of evolution are interacting and shaping the distributions of traits and genetics that we see in wild populations. Nonetheless, we often observe a reasonable amount of variation both within and among populations. So I'm showing you some photographs here of Stigmatopora nigra, which is one of the species found here in New Zealand. And each of these two photos show two different populations. And we can see that both within each of these populations and between the two populations, there's a large variation in female body size, in female body width, and in the different color patterns of these individuals. And on the right, I'm showing you here variation in Signathus scavelli, which is the species I'm going to be talking about the most during this talk. And of course, it's not just traits that include variation, right? We also know behaviors can be highly variable as well. So here's an image or a video of Stigmatopora nigra. There's a male there wiggling at a female, trying to court her, and she's displaying her trait back at him, and swimming around are a bunch of other fish that are just not at all interested, right? So a wide variety of behaviors captured in this short video clip. So the question becomes, what role does selection actually play in facilitating and maintaining this variation? We can start to address this question by using genetic tools. So we can compare genetic variation within populations, represented here by these little drawings of seahorses that have different colors. So the green ones have green genetics, purple ones have purple genetics, and we see different frequencies of these two different traits and of their corresponding genotypes across different populations. And we can look at analogous traits in a different species and compare variation between the two species as well. Today I'm going to be focusing on variation within and among populations, but focusing on only one species. Stay tuned for the next Sing Bio where I start to look across species. So how do we actually detect these processes using genome-wide data? Well, we can look across the entire genome and look for signatures of selection by investigating population differentiation, often measured by FST, and by looking at genetic diversity. And we can identify specific patterns that we expect, that we know are signatures of selection having acted on that region of the genome. So if we have a hard selective sweep, we reduce variation within the population. And if the selection is different in different populations, we expect high amounts of population differentiation. Balancing selection, on the other hand, which maintains variation, will show an increase relative to the background amount of genetic diversity and a decrease in population differentiation. So we can start to investigate these different signatures of selection. I'm going to be talking to you about work I've done in the Gulf pipefish, which is a good model for studying selective versus neutral processes. Like most cygnathids, they have limited dispersal, so migration is unlikely to be homogenizing populations. They are relatively poor swimmers, and because they have internal brooding of the embryos, they are less likely to have a larger distance of dispersal than other marine organisms. We also know that selection is acting on these fish by creating through selective pressure to be camouflaged so that they can avoid predation. And so we might expect populations that exist in different types of habitats with different characteristics might be experiencing different forms of selection to and therefore be selected for different trait optima. And we also know that this species experiences very strong sexual selection. So in the laboratory, in mesocosms in the laboratory, we were able to figure out the strength of sexual selection acting on the female bands, which are shown here on the left. And we were able to investigate the estimate of fitness as measured by reproductive success, so the total number of offspring mothered by different females. And we were able to identify that that females with very many bands, so large number of bands, or a large total area of the bands, had a higher fitness than those with fewer bands and smaller bands. So we know that sexual selection is driving this, this trait to evolve. We sampled, we also wanted to look across multiple populations to try to understand the selective pressures that might be differentiating between these different populations. And so we sampled 12 marine populations and 4 separate freshwater populations. The freshwater populations here are highlighted by the blue boxes. And I want to note that these freshwater populations were in either lakes, and so we're totally physically separated from the ocean, or they were in bays, which we would expect to experience more migration. We used genome-wide data to look at population differentiation, and using a demographic model based on those genetic data, we were able to reveal an important role for migration in these populations. So these are diagrams of the expected demographic history of the freshwater populations compared to their nearest saltwater population. And we found that in the 2 populations that were found in those isolated lakes, we found an important role of historical migration between the freshwater and saltwater populations. And in the bays, we also detected an important role of genetic drift due to a change in population size, in addition to the importance of migration. So we know that there was at least historical migration linking our freshwater populations to our saltwater populations. Nonetheless, we also found that our freshwater populations are genetically differentiated from saltwater populations. So our saltwater populations are represented on these principal component analysis in principal component space in the purple and green colors, and we can see that these marine populations cluster primarily based on geography, where all of these dark green ones are found on the same coast of Florida. The dark purple ones are the ones from the coast of Texas. And we also have the Gulf Coast of Florida represented here in this sort of pinkish light purple color. The freshwater populations mostly cluster near their geographic neighbors. And we can see this better actually in the comparison of the 3rd and 4th principal components axes, where our Texas freshwater population is here near Texas. Our Alabama freshwater population and our Louisiana ones are over here near the Alabama marine population, and the Florida population is near the Florida populations. However, we do see that there's strong differentiation, especially in the first 2 axes, between the Florida freshwater population and all of the other populations. So there's strong differentiation between the freshwater populations and the saltwater populations, but also between the freshwater populations themselves. However, we did not observe the same clustering of phenotypes. And so this again is the principal components analysis space, only this time it's for traits. And so these are linear measurements of male body traits, so body size measures essentially, the female body size measures, and then measurements of just those 2 female band traits. And we found that there's essentially no clustering, no clear patterns in these data and in terms of the traits. And so clearly this indicates to us that those important non-selective effects that were driving much of the population differentiation are not acting on the phenotypes. So some other process is impacting these different traits. Although we found a strong important effect of migration and genetic drift, We also found some clear signatures of selection, and these were distributed genome-wide. So here I'm going to be showing you a plot where on the x-axis are chromosomes and locations on chromosomes, and on the y-axis are FST values, so those measures of population differentiation. And we have these comparisons for the pairwise freshwater to its nearest saltwater neighbor estimates here. And if we look at this, the first thing that'll pop out is that there's a lot of color, right? So there are a lot of these outliers. So these ones that are popping up beyond the background level of population differentiation. And the ones that are marked with the asterisks are ones that are shared as outliers among these different population comparisons. So these are loci that are signatures of that parallel adaptation to freshwater. And 25 of these were significantly correlated with salinity, and some of them were in genes that in other fish have been shown to be associated with salinity tolerances. So among these many populations of pipefish, we were able to find that there are neutral processes acting, but we also found that signature for selection. However, that doesn't actually get us to understanding what type of selection is actually acting in these populations, and whether it's related to viability selection, so the ability to survive, eat, avoid predation, or whether it's related to sexual selection, which again, we know is very strong in this species, right? So that ability to compete for access to mates, the ability to court individuals of the other sex. And so what we can do is we know that these different types of selection act at different stages in the life cycle of our individuals. So we can compare the allele frequencies in our zygotes to the allele frequencies in adults to be able to start to differentiate. So we can compare allele frequencies among individuals from within one single population to start to differentiate between these different types of selection. And this approach is called selection components analysis. And I conducted this in pipefish. So I sampled from a single seagrass bed, a total of 225 adults. And from the pregnant males, I was able to genotype 130 embryos. Each one of these embryos, I knew specifically who the father was. And so I was able to look into both sexual selection and sex-biased viability selection by comparing the adult females that I captured to inferred successful female alleles, which I was able to create using those embryos that I sampled from the pregnant males. So basically what I could do is get a distribution of allele frequencies for all of the successful females by looking at every single genotype in the embryos and asking and determining whether I could identify which allele the mother must have contributed. So in this toy example, if the embryo had a genotype TT and the father had a genotype CT, I would know that the mother had to have contributed a T at that locus. And therefore, I could get a distribution of female allele frequencies for those successful females. And then I compared the adult females to the adult males to get that estimate of sex-biased viability selection. So again, I'm showing you a plot where we have chromosomes on the x-axis and FST on the y-axis. This time that FST value is looking at differentiation between groups from within the single population. So these are the results for sexual selection, where we have compared the females to those inferred maternal alleles. And what we can see is that the outliers, which are colored in green, are found genome-wide. Nearly every chromosome has at least one of these outliers. So there's no clear particular chromosome that seems to have aggregated these sexually selected genes. And similarly, sex-biased viability selection is also genome-wide, but it's primarily a different pattern, a different loci are involved. So ones that were found to be outliers in both of these are colored in blue asterisks, the blue stars, we can see that there are not that many of them, and in the sex bias viability analysis, there are far more unique signatures of selection. One thing you might have noticed is that these FST values are really quite large, and so these are changes in allele frequencies that occur within a single generation. And another group of researchers had created a model to try to estimate the strength of selection that might cause this level of particularly this intersexual, the male-female FST value. They estimated that surprisingly strong sex-specific selection with selection coefficients on the order of 0.75 to 0.9 being the likely amount of strength of selection that would drive this, and that seems highly unrealistic to be describing this, so something else must be going on. But these could also be indicative of unresolved sexual conflict in addition to sexual selection. And so one way that sexual conflict can be resolved is through the evolution of sex-biased gene expression. So what I'm going to show you here are some very preliminary results. So I'm excited to be able to share this with you, but yes, keep in mind it's very preliminary. I'm able to use gene expression data from 4 different tissue types, so from gonads, liver, brain, and skin, and compare expression in males and females in each of these different tissue types. And look for the distribution of male and female biased genes. So these are genes that are overexpressed in males versus females. And what I found was that we observed more genes that were overexpressed in males than in females. So those are the ones colored in blue. And I was then able to take these estimates of sex bias and compare it to those estimates of male-female FST values. What I found was that the genes that have the highest FST values have little to no sex bias in their expression, which means that males and females cannot evade any negative impacts of that particular allele through sex bias gene expression. And so this might mean that those high FST values are a signature of ongoing sexual conflict. So as I said, very preliminary. There's much more work to be done here, much more to understand, but it's exciting because now we're seeing that we have both sexual selection and likely sexual conflict interacting to drive high differentiation within a single generation between males and females. So moving forward, not only am I developing those analyses further, but I'm also beginning comparative work across multiple species of pipefish to try to disentangle those effects of sexual selection and sexual conflict. And I want to put in a little plug, currently recruiting a PhD student, ideally someone who's already in Australia or New Zealand, but I'm open to anyone who is excited about this and able to come do a PhD with me. QR code to find out more information about applying for that. So what I've shown to you today is my work to try to understand selection's role in the evolution of pipefish. And I've shown you that the Signathus scavelli populations experience a combination of genetic drift and migration, but that this is not sufficient to explain the phenotypic variation we observed. I also showed you that they've experienced local adaptation, particularly in those freshwater populations. And we have genome-wide signatures of sexual selection and sex-biased viability selection, which seem to be indicative of ongoing sexual conflict. And so with that, I will wrap up my talk. I'm eager to answer your questions, and I'm excited to hear how the rest of you are using genomic data to understand these weird and wild creatures that we are all excited about studying. Thank you very much.
Okay, thank you, Sarah. Does anyone have questions? You can unmute yourself, and ask.
Hi, Sarah. This is Tony. I have a question about first of all, the talk was incredible. I like the diversity of different approaches that you've taken. I'm curious whether that interesting bias that you saw towards male bias differences in gene expression, you looked at a series of different tissues. Was there any skew in terms of what tissues were showing this difference in expression?
Yes, there's definitely variation among the tissues. I haven't had a chance to dive into those data as fully as I'd like yet. But one thing that might be a confounding factor in this is that we actually have samples of both pregnant males and non-pregnant males. And so that there are actually more males in the data set than females. And so I'm still working to try to figure out how to get a better balance of that. But yes, certainly some of the biggest sexual dimorphism is in the gonads, unsurprisingly, although the liver is also quite sexually dimorphic.
Thanks.
Thank you. Does anyone else have questions? Or you can put your questions in the chat box. Okay. Hi, Sarah. I have a small question about the differentiation between the saltwater and freshwater population. Have you found any genes in the differentiation between population?
Yeah. So you're asking about the genes that might be differentiated?
Mm.
Yeah, so some of those SNPs are in coding regions of the genome, and some of the coding regions have been shown in other fish species to be associated with saltwater tolerance and other physiological aspects that would be relevant to moving into freshwater, but not all of the SNPs are in coding regions. And, of course, with all of this type of genome scan, it's very correlative. So there needs to be, further work done to try to understand exactly what genes and what traits are being locally adapted in those populations.
Okay, thank you.